Hello, dear my students. Today our fifth lecture. We continue our lecture in online. About today our fifth lecture, but topic the carbohydrates and nucleic acids. As you know, my name is Kamal Bədəlova. I'm assistant professor of the General Toxicological Chemistry Department. My lecture for my dear students of the pharmacy faculty for the second course. Okay, at first we will study our carbohydrates. After it, we will go to the nucleic acids. Okay, what is carbohydrates? The carbohydrates, every time we try carbohydrates in our foods. For example, we eat sugar, we use sugar, we use vegetables, we eat fruits, and another, as you see in our slide. About the definition, what is carbohydrates? Carbohydrates are polyhydroxyaldehydes or the polyhydroxyl ketones or compounds that give polyhydroxyaldehydes or polyhydroxyl ketones on hydrolysis. Carbohydrates are aldehyde or ketone derivatives of polyhydric alcohols. Which classification? You know that carbohydrates can be classified into monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the smallest carbohydrates. They can't be hydrolyzed into smaller carbohydrates made up the carbon, hydrogen, and oxygen. Have the general formula CnH2n or N. Disaccharides are monosaccharides, but you know that our carbohydrates, they are biopolymers, but for the biopolymers, as for all polymers, we have monomers. And as carbohydrates, they are polymers, but monosaccharides, they are polymers for the disaccharides and the polysaccharides. Okay, the constituent monosaccharides may be identical or the different. The common disaccharides are sucrose, lactose, and maltose. Monosaccharides and disaccharides are called sugars because of their sweet taste. Polysaccharides are made up of a large number of monosaccharide molecules. They are monomers. Monosaccharides are the monomers for the polysaccharides. Those having 3-6 monosaccharide units are called oligosaccharides. Those having more than 6 monosaccharide units are called polysaccharides. About the function of carbohydrates, which function are they? Main source of energy, spare protein from being burnt so it can be used to build and repair. Dietary fiber can help lower blood cholesterol, part of connective tissue. Some are hormones and enzymes and genetic material. We thought that we can classify our carbohydrates to monosaccharides, disaccharides, and the polysaccharides. Okay, now we'll study about the monosaccharides. We have some classification of the monosaccharides. They classify to aldoses, those that contain an aldehyde group; ketoses, those that contain a ketone group; trioses, they have 3 carbon atoms; tetroses have 4 carbon atoms; pentoses have 5 carbon atoms, and so. Okay, on according the number of carbon atoms, we can classify our monosaccharide to trioses, tetroses, pentoses, and hexoses. Also, we can continue this list. And an aldohexose is an aldehyde sugar with 6 carbon atoms. aldohexose is an aldehyde sugar with 6 carbon atoms. The os ending indicates that the molecule being named is a carbohydrate. Okay, this is about the stereoisomers of the carbohydrates. You know that we have the mirror image. At first, we will pick what is stereoisomers. Carbohydrates contain a chiral carbon atom and have stereoisomers. My dear students, maybe remember what is chiral carbon atom. Chiral carbon atom is a carbon atom binding with 4 different groups or the atoms. Enantiomers are represented using a Fischer projection. In Fischer projections, chiral carbon atoms sit at the intersection of a vertical line and a horizontal line. The horizontal lines represent bonds pointing towards the weaver, and the vertical lines are 4 bonds pointing away from the weaver. For monosaccharides, Fischer projections are drawn with the carbon atoms running vertically and the aldehyde or ketone group or at or near the top. Okay, in this figure you can see, in this slide you can see some figure where we have the mirror image of 2 trioses. Okay, they are trioses D-glyceryl aldehyde and L-glyceryl aldehyde. Okay, chiral carbon atom gives rise to stereoisomers. What is chiral carbon atom? Chiral carbon atom is asymmetric carbon atom. I told in last slide. This is a carbon atom with 4 different functional groups or the atoms. For example, this carbon atom binded with 4 different functional groups or atoms: hydrogen, OH group, CH2OH, and the COO is aldehyde group COH. Okay, the name of this compound glyceryl aldehyde. But what is this? How many carbon atoms we have in glyceryl aldehyde? Just 3 carbon atoms, because this compound related to the trioses with 3 carbon atoms, carbohydrate with 3 carbon atoms. But this is not sugar. Mirror image. What is mirror image? Mirror image molecules are named as enantiomers. Okay, but near carbon atom, near asymmetric carbon atom, we can see hydrogen atom and OH group. If OH group, hydroxyl group, in right side, this is D isomer. But if OH group in left side, this is L isomer. Just in first slide you saw, when drawn in a Fischer projection, the OH attached to the chiral carbon atom next to the last carbon atom, just above the last carbon atom. You know that for the glyceryl aldehyde we have just 1 asymmetric carbon atom. With 1 asymmetric carbon atom we can define D or L isomers very easy. But when we have 5 or 6 carbon atoms in our carbohydrate, for example, for the hexose, we have just 4 asymmetric carbon atoms. Just last carbon atom, just last asymmetric carbon atom, we can take for the define D or L isomers. Okay, because when drawn in a Fischer projection, the OH attached to the chiral carbon atom next to the last carbon atom, just above the last carbon atom, points to the right in a D monosaccharide and to the left in an L monosaccharide. In nature, most monosaccharides are D sugars. So D-glyceraldehyde is commonly found while L-glyceraldehyde is not. Okay, in this figure we can see the D and L isomers of aldohexose. This is glucose, D-glucose and L-glucose. But what is D-glucose? You can see fifth carbon atom. This is last asymmetric carbon atom. Last carbon atom, this is carbon atom of CH2OH. But last asymmetric carbon atom, fifth carbon atom. If OH group of the last asymmetric carbon atom in the right side, this is D-glucose, this D isomer. But if OH group in the left side, this is L isomer. Okay, D-glucose, L-glucose, D-galactose in the right side. And fructose. What is fructose? Fructose also, this is ketohexose, where we have 6 carbon atoms, but the ketone group. And ribose, deoxyribose, they are pentoses, where we have just 5 carbon atoms. In our figure, it writes aldohexose stereoisomers, aldohexoses have 4 chiral carbon atoms and exist as 16 stereoisomers. How can we calculate the number of the stereoisomers for our carbohydrates with the asymmetric carbon atoms? Okay, 2 in 4 degree. At first, how many stereo asymmetric carbon atoms has, for example, glucose? Let's take our glucose. This is aldohexose. Okay, in our compound, in our glucose, both D-glucose and L-glucose, we have just 4 asymmetric carbon atoms. By formula 2 in n degree, what is n? N, the number of the asymmetric carbon atoms. Okay, but for the glucose, for example, for glucose, for galactose, 2 in 4 degree equal 16 stereoisomers. Just by this formula, my dear student, you must remember this rule. 2 in n degree is formula for the calculation of number of the stereoisomers. Just by this way, we can find how many stereoisomers has this carbohydrate. For example, for the ribose, how many asymmetric carbon atoms has ribose? 1, 2, 3. For the deoxyribose, you can search also how many asymmetric carbon atoms has our pentoses and another carbohydrates. In our slide, we have the configuration of the aldoses, of D-aldoses. This is triose, tetroses, pentoses, and hexoses. The list of the D-aldoses. What is aldose? You remember aldose, the carbohydrates with the aldehyde group. Ketose has one less asymmetric carbon atom than aldose. Therefore, they have few stereoisomers. Why they have less than one? Because they contain carbonyl group. Okay, the dihydroxyacetone, D-erythrulose, D-ribulose, D-xylose. How many carbon atoms here? 1, 2, 3, 4. 1, 2, 3, 4, 5. It means this is pentaketose, trioketose, tetraketose, and hexaketosis, or the ketohexoses. What is ketohexoses? With 6 carbon atoms and with keto group. Glucose, what is glucose? Glucose blood sugar, the main fuel molecule for our bodies. Fructose sugar from fruit, galactose sugar found in lactose in milk, ribose a composition of RNA and DNA, they are nucleic acid, and deoxyribose a composition of DNA. Important monosaccharides, which monosaccharides? D-glucose is also known as blood sugar. One form of energy storage in the body involves combining glucose molecules to produce glycogen. D-galactose is combined with glucose to produce lactose, a disaccharide that gives milk its sweetness. D-fructose or fruit sugar is a ketose found most often in nature. Fructose plays a key role in energy production. Pentoses and hexoses are also important monosaccharides. Other most abundant monosaccharides found in nature. D-ribose and D2-deoxyribose are aldopentoses that are often incorporated into large biomolecules including NAD pH, NAD+, and the nucleic acids RNA and DNA. The structural difference between these two monosaccharides is that 2-deoxyribose is our second carbon atom. Near second carbon atom, we can see just 2 hydrogen atoms in deoxyribose, but in ribose we have OH group. Okay, is that 2-deoxyribose lacks an OH group at second carbon atom, hence 2-deoxy part of the name. What is deoxy? It means that we removed one oxygen atom from ribose and we form deoxyribose. Because the formula of the ribose C5H10O5, but the formula of the deoxyribose C5H10O4. Some examples of other kinds of monosaccharides or the sugar. This fructose, L-rhamnose, D-glucosamine. D-glucosamine, they contain NH2 group at the second carbon atom, N-acetyl D-glucosamine, N-acetyl D-glucosamine, D-gluconic acid, and D-glucuronic acid. About the cyclic hemacetal formation, when the carbonyl group of an aldehyde or ketone reacts with the hydroxyl group of an alcohol, a hemacetal forms. In D-glucose, the fifth OH group reacts with the carbonyl group of the first carbon atom. Okay, here we can see fifth carbon atom, the OH group of the fifth carbon atom, and carbonyl group of the first carbon atom. Between them we can write the cyclic hemiacetal, this hemiacetal carbon atom. This carbon atom named as hemiacetal carbon atom. About the cyclic form of the glucose, this is pyranose ring, this is furanose ring. About the mutation is the interconversion of the alpha and beta anomers. Okay, now I will explain what is alpha and beta anomer. In this is our open form of D-glucose. You can see here aldehyde group. From open form we can form alpha glucopyranose, this glucopyranose, and the D-glucopyranose. What is alpha glucopyranose? But if OH group in down This is alpha anomer. But if OH group in up, this is beta anomer. Usually we can name alpha form. This is cis. This is trans isomer. Why? Because the direction of OH group and CH2OH group, they are opposite. Because this is trans form. But cis form, why? Because the directions of OH group and CH2OH are in the same site. This is cis form. Okay, these two forms of glucose are said to be anomers, alpha and beta anomers. And down you can read for my dear student. For example, glucose forms a pair of cyclic hemiacetals when the OH group attached to carbon atom 5 reacts with the carbonyl group. The cyclic hemacetal with the OH group on carbon atom pointing down is the alpha anomer, and that with the OH group pointing up is the beta anomer. Monosaccharides that form anomer containing 6-membered rings are called pyranoses. About the anomers, I told that the one with the hemiacetal OH group on carbon, first carbon, pointing down is called the alpha anomer, and up, this is beta anomer. Okay, anomer, and that with the OH group pointing up, this is beta anomer. This is beta, this is alpha anomer. OH group and OH group up. Cyclic forms of the fructose ribose and deoxyribose. The cyclic forms of the this is alpha fructose or alpha D fructofuranose, alpha D ribofuranose or just alpha D ribose, alpha D 2-deoxyfuranose or the just alpha D 2-deoxyribose. Disaccharides. What is disaccharides? Disaccharides are sugars which contain two monosaccharide residues. About examples, the maltose, 2-glucose, alpha 1,4 bond, cellulose, 2 glucoses which are formed by the beta 1,4 bond. This is what is 1, what is 4. In next slide I will show you. They are linkage between two glucoses. Lactose, glucose bind by the beta 1,4 bond. Sucrose, glucose and fructose. Okay, maltose contain 2 glucose molecules, 2 alpha glucose molecules. Cellulose, 2 beta glucose molecules. Lactose contain galactose and the beta glucose molecules. Sucrose contain glucose and fructose molecules. Okay, they are our maltose molecule. You can see here alpha 1,4 glycosidic bond. Okay, what is alpha 1,4 glycosidic bond? OH between OH group of the first carbon atom. At this carbon atom, this anomeric carbon atom, between OH group of the first carbon atom and the first monosaccharide and the fourth OH group of the second monosaccharide, we can form disaccharide by the formation of alpha 1,4 glycosidic bond. Okay, the disaccharide maltose is an acetyl glycoside formed by the combination of 2 D-glucose molecules. An alpha 1,4 glycosidic bond connects the two glucose residues. this is lactose, and this formula of the lactose. In lactose, we can say beta 1 for glycosidic bond. But in sucrose, we can see alpha beta 1 2 glycosidic bond. But if and one but in our maltose, near the anomeric carbon atom of the second monosaccharide, we can see the free OH. In molecule of the lactose, same, we can see OH free OH group at the anomeric carbon atom of the second monosaccharide. In cellulose, same, we can see OH group, free OH group at the anomeric carbon atom of the second monosaccharide. But this difference near sucrose, in our sucrose molecule, we can't see any free OH group near the anomeric carbon atom. And because sucrose, the disaccharide which we can't reduce. But cellulose, lactose, and maltose, they are disaccharides which we can reduce. They are glycolipids. For example, can you spot the sphingosine and the fatty acyl group? Okay, the compounds which contain D-galactose residue, the N-acetyl acid residue, N-acetyl D-galactose amine residue, D-galactose residue, D-glucose residue, where glucose glycolipids. These compounds often consist of the fatty acid residue and the glucose or the galactose residue attached to a sphingosine backbone, the glycolipids. But which role of the disaccharides? What about maltose? Form the constituent of the building blocks of amylose, amylopectin, and glycogen, used for energy. Amylose, amylopectin, and glycogen, they are polysaccharides. Cellulose forms the constituent or the building block of cellulose, also polysaccharide, leading to structural role for not energy role. Galactose, milk sugar, sucrose, table sugar, has an energy role. About the polysaccharides, the carbohydrates that contain many monosaccharide residues linked together are called monosaccharides. In living things, some polysaccharides help provide structural, and some act as energy storage molecules. We'll study these polysaccharides just: starch, which consists of amylose and amylopectin, glycogen is cellulose, chitin, chondroitin sulfide, and the hyaluronic acid. Polysaccharides in plants. Which polysaccharide we have in starch is produced by plants as a way to store energy. Starch consists of two different polysaccharides, amylose and the amylopectin, each of which is composed entirely of maltose or glucoses, building blocks. Amylose, single long chain of glucoses, no branching. Amylopectin, one long main chain of glucose attached to branching. Okay, we have two differences between amylose and amylopectin. Amylose without any branch, not branching compound, single long chain of glucose, but amylopectin also contain molecules of the glucose, but they are branched molecules. Every branchings every 25-30 glucose residues in animal or the human. Also, we have another difference between amylose and amylopectin. If amylose we can dissolve in water, but amylopectin we can't dissolve in water. In animals or the human, glycogen and energy storage polysaccharide in animals and human, not in plants. The structure of glycogen looks like that of amylopectin, except it is more branched, every 8-12 glucose residues. Okay, this is structure of the amylose. We can see that just continue chain, but amylopectin has the branch. But if in amylose structure we can see just one alpha 1,4 glycosidic bond, but in amylopectin we can see two bonds, 1,4 glycosidic bond and alpha 1,6 glycosidic bond. And this is structure of the starch. But amylopectin or the glycogen, the branch is the one on top. About the cellulose is of major importance to structure of plants. Wood is about 50% cellulose because it provides support to stems and stalks and provides a touch water insoluble protective barrier. Most animals don't produce enzymes related to hydrolysis cellulose intercellular and individual glucose molecules to cellulose is not a good animal food source. Bacteria found in the digestive tracts of horse, cows, and termites contain cellulases enzymes that break down the cellulose which is digested. Chitin makes up the exoskeleton of crustaceans and the insects and is present in the cell walls of some algae and the fungi. The structure of the cellulose, here we can't see any branch, it's just a simple polysaccharide, with a simple monosaccharide which hasn't any branch, just the continuous chain. A structure of the chitin without also any branch, but chitin usually we can classify polysaccharide to the homopolysaccharide and heteropolysaccharide. What is homopolysaccharide? Homopolysaccharide, the compounds which contain two same or the same units of the monosaccharides. But what is heteropolysaccharide? Heteropolysaccharide, the compounds which contain different monosaccharides units. But our chitin, we can relate the chitin to the amino sugars. Why? Because in our structure of the chitin, you can see amino group. This is N-acetyl D-glucosamine residues. From the other monosaccharides, which other monosaccharides we know? The hyaluronic acid found to be important component of the representing fluid that surrounds the joints in animals or the human body and in the vitreous humor-clear gel inside the eye. It is made up of alternating residues of N-acetyl D-glucosamine and D-glucuronate. Okay, it means that our hyaluronic acid, heparin, chondroitin 4-sulfate, They are related to the heteropolysaccharides, which contain different monosaccharide units. Heparin is a natural anticoagulant substance. It inhibits blood clotting mechanisms. Chondroitin phosphate consists of N-acetyl D-galactosamine phosphate residues alternating with D-glucuronate residues. These heteropolysaccharides are present in cartilage, tendons, and other connective tissues. Contains nitrogen and the sulfate. This is structure of our acid, hyaluronic acid. The structure of the chondroitin sulfate, they are heteropolysaccharides. And the structure of heparin, which consists of D-glucuronate 2 sulfate and sulfur D-glucosamine 6 sulfate. Okay, in this slide we finished with the carbohydrates. Now we go to the nucleic acid. What is nucleic acids? Nucleic acids, we know that we have two nucleic acids, DNA and RNA. They are long chains of the repeated nucleotides. Okay, DNA and RNA are nucleic acids. They are biopolymers. About the nucleotide, what is a nucleotide? A nucleotide consists of first nitrogenous base, pentose sugar, and one or more phosphate groups. They are nucleotide. Okay, bases attached to a sugar is called nucleoside. Sugar with phosphate and with base, this is nucleotide. Okay, from the DNA has only thymine, 2-deoxyribose. Which difference we have between the DNA and RNA? In DNA and in RNA, in both we have which bases? Adenine, guanine, and cytosine. But in RNA, just uracil ribose, and in DNA we have thymine and 2-deoxyribose. Ribose related to RNA, deoxyribose to DNA, because from name we can say ribonucleic acid and deoxyribonucleic acid. In this slide also we'll repeat what is nucleic acid. They are polymers that consist of nucleotide residues. Each nucleotide is put together from 3 building blocks: phosphoric acid, there are our phosphoric acid residue, monosaccharide, there are our sugars, and the organic bases, there are our organic bases. For example, adenine, cytosine, guanine, thymine, uracil, they are organic bases. About the phosphoric acid, at low pH present as the phosphoric acid. At the high pH, it present as a phosphate ion, but in physiological pH 7.4 present as the dihydrogen sulfate ion and hydrogen phosphate ion. About the monosaccharides, all nucleotides are constructed from one of these two monosaccharides, ribofuranose, beta-ribofuranose, and beta-2-deoxyfuranose. During explanation of the monosaccharides, I explained that we have alpha form and the beta alpha anomer and beta anomer. What is beta anomer? If OH group pointing up, they are beta anomer. And about the organic bases, we have thymine, guanine, cytosine, and adenine. Okay, in this slide also we have about the nucleotides. The linkage of the phosphate group, sugar, and the base. Sugar, they are monosaccharide. Okay, this is our nitrogenous base. For example, adenine, sugar, and the phosphate group. About sugars, I told in in another slide, sugars, they are ribose and the deoxyribose. Okay, you can see that in ribose we have the additional oxygen atom and the second carbon atom, but in deoxyribose we haven't any oxygen atom near the hydrogen at the second carbon atom. Which nitrogenous bases, the derivatives of which nitrogenous bases we can include to the RNA and DNA. They are purine and pyrimidine. As you see, pyrimidine, this is six-membered heterocyclic compound with two heteroatoms, which heteroatoms they are nitrogen atoms, and the molecule of the purine, this is fused heterocyclic compound which contain two heterocycles. First part, this is pyrimidine ring, and second part, this is imidazole ring. Purines, in both DNA and RNA we have adenine and guanine. They are derivatives of the purine. But maybe you can meet with the question which purines rings we have in DNA and RNA. They are adenine and the guanine. Adenine and guanine. Pyrimidines, from the pyrimidines ring we know cytosine, uracil, and the thymine. Just cytosine we have in both in RNA and DNA. In RNA we have uracil, in DNA we have thymine. This is nomenclature of the nucleotides and the nucleosides. Possible bases are cytosine, thymine, and uracil. They are derivatives of the pyrimidines, and purines, they are adenine and the guanine. They are bases, but monosaccharides, ribose or the deoxyribose, together ribose or the deoxyribose with bases, they are nucleoside, and nucleoside plus phosphate we can form nucleotide. In this slide you can see what is nucleoside, what is nucleotide, and all of them. This is nucleotide. For example, nucleosides, they are monosaccharide with the base and plus phosphate. This is nucleotide. In the sugar, if X, this is OH, this is ribose, but if X is hydrogen, this is deoxyribose. Base, for example, adenine, guanine, thymine, cytosine, uracil, this depends. From the purines in this table, we can see which for the adenine, for the guanine, for purines and for pyrimidines derivatives, which derivatives we have in our compound. For example, nucleoside RNA, deoxynucleoside, this is DNA, and base plus sugar. With base and sugar, the name of this nucleoside. For example, if we have adenine in our compound, this is adenosine, deoxyadenosine. Guanosine, deoxyguanosine. This is for the DNA. Titidine, deoxytitidine. Uridine, deoxythymidine. For the nucleotide which contain base, sugar, and the phosphate group. For example, if our compound contain adenine, we can name as adenolate, deoxyadenolate. For the guanine, guanylate, deoxyguanylate. For the cytosine, titillate, deoxytitillate. For the uracil, uridylate, deoxythymidylate, uridylate or the deoxythymidylate. They are first part for the ribonucleic acid, and second part, for example, deoxy, they are for the DNA. for the deoxyribonucleic acid. DNA deoxyribonucleic acid was firstly isolated by the Swiss physician Friedrich Mescher in the 1869 year. Actually, it was Rosalind Franklin's discovery, X-ray diffraction picture of structure of DNA that led to Crick and Watson double helix model in 1953. She died from cancer at the age of 37 in 1958. Professor Maurice Wilkins received the Nobel Prize with Crick and Watson for three-dimensional structure of DNA in 1962. The polymeric structure of DNA may be described in terms of monomeric units if increasing complexity. The three relatively simple components are phosphoric acid, purine or the pyrimidine base, and 2-deoxyribose. They are components of the DNA. Okay, from the pyrimidine bases, this is cytosine and thymine. Purine bases, adenine and guanine. This is a series repetition of our last slides. And examples, which examples of the modified bases found in the transport RNA? 1-methyl adenine, 7-methyl guanine. They are modified bases. And another two examples are 3-methyl cytosine and pseudouracil. And what is a deoxyribonucleotide? This is 2-deoxyadenosine 5'-phosphate. Why adenosine? Because our compound contains base adenine. And deoxyribonucleotide, which contains cytosine base, this is 2'-deoxytitidine 5'-phosphate. 5'-phosphate. Deoxyribonucleotide, which contains thymine, thymidine 5'-phosphate. Types, which types of bonds we have in our compounds? Okay, type of bonds in 2'-deoxytitidine 5'-diphosphate. And in our compound you can see also beta N glycosidic bond, but this bond named as beta N glycosidic bond. Why N glycosidic bond? Just through a nitrogen atom we can write this bond here. This is phosphoric anhydride bond. This is phosphoric anhydride bond. This is phosphoric ester bond. What is ester? Maybe you remember, dear my students, between acid and the OH group we can write ester bond. About the nucleoside D and 3-phosphates. Nucleoside D and 3-phosphate are energy-rich compounds that release large amounts of energy when the phosphates are removed hydrolyzed. Adding a phosphate to a nucleoside monophosphate generates a nucleoside D-phosphate. This is AMP. What is AMP? This is adenosine monophosphate. Adenosine 5' monophosphate. And additional phosphate group will generate nucleoside D-phosphate. Example, this is adenosine 5' D-phosphate, ADP. Adding a phosphate to the nucleoside D-phosphate generates a nucleoside 3-phosphate, adenosine 5 prime 3 phosphate. This is ATP. You know that ATP a good source energy in our organism and in for the living organism. This is nucleoside D and 3 phosphates. This ATP, such as adenosine 5 prime 3 phosphate ATP, as the immediate source of energy for most of the energy regain processes that take place in living things. This is the picture which describe our ATP and ATP energy cycle. Energy from sunlight or from food to our organism, to living organism, and this energy will go to the will used for the biochemical processes in our organisms. But from these processes, the energy available for the cellular work and for the chemical synthesis. Okay, ATP is ATP. This is ADP, where we have just 2 phosphate groups. Cyclic nucleotide. What is cyclic nucleotide? Cyclic nucleotides are formed when the one phosphate group has 2 phosphoester connections to the same nucleoside monosaccharide. This is cyclic form. Adenosine cyclic monophosphate, CAMP. Adenosine cyclic monophosphate. In most cases, they functions as effectors for allosteric enzymes to regulate biological processes. Okay, there we can see the names of DNA base derivatives. This table describes the names of DNA bases derivatives. Base sequence is read from the 5 prime end to the 3 prime end. About the DNA structure, the structure of DNA is understood in terms of 3 levels of structure: primary structure, secondary structure, and the tertiary structure. Every cell nucleus in a particular living organism contains the exact same DNA molecules. Human DNA contains 3 million paired deoxyribonucleotide residues and contains 25,000 genes. What is genes? Genes, they are stretched of DNA that carry the codes for protein production. But the primary structure of a nucleic acid refers to the sequence of its nucleotide residues. Secondary structure pertains in DNA to the helix formed by 2 interactions of 2 DNA strands. This is DNA molecules. This is our secondary structure. The 2 DNA strands are held to one another by base pairing, hydrogen bonding between the bases attached to the sugar phosphate backbone. Tertiary structure for nucleic acid. Tertiary structure refers to the overall 3-dimensional shape. In DNA, tertiary structure arises from supercoiling, which involves double helices being twisted into more compact shapes. RNA are easily hydrolyzed under mild alkaline conditions to nucleotides, which is cleaved in alkaline medium, the phosphoric acid, to form nucleosides that are hydrolyzed in acidic medium to the heterocyclic base and the sugar. The secondary structure of DNA is determined by the spatial organization of the polynucleotide chain. The orientation of the heterocyclic base on each nucleotide residue is perpendicular to the axis of the double helix. Okay, here Watson and Crick with their model of the DNA in this track. According to the Watson-Crick model of a DNA molecule, consists of two polynucleotide chains, forming a double helix with diameter of 1.82 nanometer. This is 20 angstrom. At each turn of the helix are 10 base pairs. 10 base pairs. The sugar phosphate backbone runs along the outside of the helix, and the amine bases hydrogen bond to one another on the inside. Both major and minor growths are visible. Two polynucleotide strands are antiparallel to each other, so direction of phosphodiester formation is opposite. One chain is 5' 3' and other of 3' and the 5' end. This is structure of this a picture of the DNA double helix fragment in spice filling and wireframe format. Base pairing between guanine and cytosine, we have three double bonds. Three hydrogen bonds. Okay, between guanine and cytosine, we have three hydrogen bonds. But between thymine and adenine, we have two hydrogen bonds. In our nucleic acid, we have one rule. This is charge of principles. Adenine, this adenine always pairs with thymine in DNA. Cytosine also pairs with guanine in DNA. The amount of adenine is equal to the amount of thymine, same for cytosine and the guanine. Reach different levels. There are different levels of DNA structure to fit the enormously long DNA duplexes into the nuclei of human cell. This is our double helix DNA. Two pairs of 4 different proteins, 150 to 200 base pairs, nucleosomes. The following we have three processes are involved in the duplication, transfer, and use of genetic information. This is replication. Replication, the process of which a replica or identical copy of DNA is made when a cell divides. This is replication. What is transcription? The process by which the genetic messages contained in DNA are read and copied. Replication, transcription. And third process is translation, the process by which the genetic messages carried by RNA are decoded and used to build proteins. Replication, in this slide we will repeat replication. What is replication? It was the process by which identical copies of DNA are made so that information can be preserved and handed down to offspring. Transcription, the process by which the genetic messages are read and carried out of the cell nucleus to ribosomes where protein synthesis occurs. Translation, the process by which the genetic messages are decoded and used to synthesize proteins. Okay, replication of DNA, transcription to RNA, and translation to the synthesis of the proteins at last. What about the structure of RNA? RNA structure is similar to DNA. Both are sugar phosphate polymers and both have nitrogen-containing bases attached, but there are 3 differences between them. RNA contains different monosaccharide residues. In RNA we have ribofuranones, beta ribofuranones, but in the DNA we have another monosaccharide. This is beta 2 deoxyfuranose. RNA contains the bases adenine, guanine, cytosine, and uracil instead of adenine, guanine, cytosine, and thymine in DNA. In RNA, uracil replaces thymine. Okay, in RNA uracil replaces thymine. Thymine in DNA, uracil in RNA. RNA exists as a single strand instead of a double strand. If in DNA we have double helix, but in RNA we haven't any double helix, just a single strand. About the function of RNA, DNA has only one kind of function, storing genetic information, and by contrast, the different kinds of RNA perform different functions. Okay, the following three RNA make it possible for the encoded information carried by DNA to be put to use in the synthesis of proteins. Their transfer RNA are the smallest of the three types, 73 to 93 nucleotide residues, and they carry the correct amino acid to the site of protein synthesis. About the messenger RNA, mRNA are of variable size depending on the protein to be manufactured and contains the information that specifies which protein should be made. This message is the sequence of RNA nucleotides that is complementary to the template strand of DNA. Ribosomal RNA are relatively long RNA strands, hundreds or thousands of nucleotide residues that combine with proteins to form ribosomes, the multi-subunit complexes in which protein synthesis takes place. And about transcription, the first step in using the information stored in DNA to produce proteins is transcription using DNA as a template to make RNA, formation of the messenger RNA. About the genetic code, what is genetic code? The ribonucleotide sequence in messenger RNA chain is like a coded sentence that specifies the order in which amino acid residues should be joined to form a protein. Each word or codon in the mRNA sentence is a series of 3 ribonucleotides that code for a specific amino acid. For example, the series uracil guanine on an mRNA chain is a codon directing incorporation of the amino acid leucine into a growing protein chain. Of the 64 possible 3-base combination in RNA, 60 code for the specific amino acids, 1 codes for chain start and 3 code for chain termination. In our table you can see this one. This is translation protein synthesis. This picture describes protein synthesis. About the transfer RNA, each transfer RNA carries an anticodon, a series of 3 bases that is complementary to a codon. This is picture of the transfer RNA. The structure of the transport RNA also. About the protein synthesis or the translation takes place in 3 steps. This is initiation, ribosome messenger RNA and the transport RNA come together to form a complex. Elongation, amino acids are joined to the growing polypeptide chain. And third step, termination. The protein has been synthesized and the ribosome messenger RNA transport RNA complex dissociates. this picture, but this picture tell us that this 0.1% DNA difference between us may have to do with the number of nucleotides in a person's DNA. If you envelop all of the DNA you have in all your cells, you could reach the moon 6,000 times. This is our about the DNA theme. About the nicotine amide adenine, this dinucleotide is one of the principal oxidation reduction reagents in biological systems. Bell's test pentose detection in products of the nucleoprotein hydrolysis when reached with concentrated solution of H2SO4 or dilute HCl pentose are hydrated to form furfural, which is condensed with orthanol 3,5-dihydroxytoluene. Okay, Bell's test also they gave red product. Okay, what is Bell's test? They gave red products of condensation with thymol 2-isopropyl 5-methylphenol. Any permanent in our also DNA and RNA nucleic acid, we have mutation. What is mutation? Any permanent change in the primary structure of sequence of nucleotide residues in DNA is called a mutation. Mutation might involve the switching of one base pair for the another, of addition the deletion of base pair, and errors in replication and exposure to mutagens as a common cause of the mutations. We have two types of the mutation. They are substitution substitution mutations, in which one base substitutes for another in the normal base sequence. One purine for another, one pyrimidine for another, and purine for a pyrimidine, or the pyrimidine for a purine. And frame shift mutations, in which a base is inserted into the normal base sequence or is deleted from it. About the importance and functions of the nucleotides, they are building blocks of DNA and RNA. We know about it. Then they are derivatives. ATP formation, which is a major source of energy in the cell. GTP, UTP, CTP, what is there, guanine, uracil, acetazyl, are sources of energy in certain metabolic pathways. What is ATP? In ATP, our nitrogenous base, this is adenine. In GTP, this is guanine. In UTP, this is uracil. In CTP, this is cetazine. They are sources of energy in certain metabolic pathways. Formation of the coenzymes NAT, NADP, FAT, and the coenzyme A. Then CAMP, this is cyclic. AMP acts as a second messenger inside the cell for many hormones, and CGP acts as a cellular mediator. Some nucleotides are important regulators for many metabolic reactions. Nucleotides act as carriers of activated intermediates such as UDP, glucose is synthesis of glycogen, CDP, choline is synthesis of the phospholipids, GDP, mannose, is in synthesis of the glycoproteins. Synthetic analogues of the naturally occurring nucleotides, these compounds inhibits the growth cancer cell by inhibiting their enzyme activity or inhibiting the synthesis of either DNA or the RNA. 5-fluorouracil. Allopurinol, a purine analog, is widely used in the treatment of gout. With our allopurinol picture. About the antiviral drugs, they are acyclovir and the zidovudin. DNA synthesis terminates whenever ester is incorporated into the growing DNA strands in the source of reverse transcription. Okay, there our new viruses are not produced. New viruses are not produced. Okay, dear my students, this is the end of our lecture. Be safe and very thanks for your attentions, my dear students. Okay, see you next lecture.